In this lesson, we continue looking at neurons by considering membrane potential. We're going to define a bunch of terms related to membrane potential, explain the concept of the electrochemical gradient, and look at how ion pumps and channels affect the membrane potential. If we were to take a look at an axon from the side, so this is meant to be a side view of an axon, and stick a voltmeter in it. Here's one lead of a voltmeter, and we always need to measure the voltage relative to something else, so we'll put the other lead outside. We would see that the inside of an axon is actually negative compared to the outside. That's what I'm drawing here. The inside is negative compared to the outside, and typically it measures about negative 70 millivolts. Now over here I have an oscilloscope tracing that's going to show what this voltmeter is reading at different times. So I said that typically the membrane potential is about negative 70 millivolts. And I'm going to draw that here as a dotted line because this is what the membrane potential would be if nothing were happening. So this is negative 70 millivolts, and we call that the resting membrane potential. I'm going to number these terms on the right here with their corresponding parts on the diagram so that you can see what we mean. So the inside of the cell is more negative than the outside of the cell, and that's achieved by the cell doing work. What I'm drawing here is the cell membrane. This is the outside of the cell. This is the inside of the cell, which we said is at negative 70 millivolts roughly at resting compared to the outside. This is the membrane here, and these are proteins embedded in the membrane. The one on the left is a sodium-potassium pump. It is going to pump 3 sodium ions out and 2 potassium ions in, and it's going to use the energy from ATP to do that. So it hydrolyzes ATP to ADP plus inorganic phosphate. That releases energy, and it uses that energy to pump 3 sodiums out and pump 2 potassiums in. Now that means that more positive ions are going out than positive ions are coming in, and that deficit is what leads to the negative 70 millivolt resting membrane potential. There are also other channels called leak channels that are always open, and for example there are potassium leak channels that will let some potassium leave from the inside to the outside of the cell, and that also contributes to the resting membrane potential. As a result of active transport by the sodium-potassium pump and contributions from these leak channels, sodium and potassium ions end up in very different concentrations on either side of the membrane. Sodium ends up in high concentration outside of the axon and in low concentration inside of the axon, and potassium the reverse. And this results in what is referred to as an electrochemical gradient for each of these ions. There are 2 reasons for which sodium would, if it were able to, would like to cross the membrane. It can't because the membrane is impermeable to ions, but if sodium were able to cross the membrane, it would like to for 2 reasons. One is purely chemical, and that's that ions and other particles tend to diffuse from areas of high concentration to areas of low concentration. So that chemical gradient would tend to move sodium in. It's in high concentration outside, low concentration inside, so it would diffuse in just like any other particle. But there's a second reason as well. Sodium is a positive ion. Remember that the inside of the axon is negative relative to the outside. Like charges repel, so there are a bunch of positive charges outside, and opposite charges attract. It's more negative inside. So for that electrical reason, sodium would like to move from the outside to the inside as well, getting away from the positive charges and getting closer to the negative charge inside the axon. We refer to that combination of the electrical effect and the chemical effect of standard diffusion as creating an electrochemical gradient. In this case, both of those things are pointing in the same direction. The concentration effect, the chemical effect, and the opposites attract effect, the electrical effect. And so there's a powerful pull for sodium into the inside of the axon. You can think of an individual sodium ion outside of the axon as being like a boulder sitting at the top of a hill. If we were to allow the boulder to roll down the hill, if we were to allow the sodium ion to cross through the membrane and get inside the axon, both of those would release energy. So the electrochemical gradient stores energy, just like the hill stores energy, in that the boulder at the top of the hill has energy once it's been pushed up there. In the membrane potential case, the pushing up the hill is happening here. The sodium-potassium pump is using energy from ATP to push sodium up the energy hill up its electrochemical gradient or against its electrochemical gradient. That's the language we use. Whereas over here, if membrane permeability for sodium was suddenly increased, sodium would fall down its electrochemical gradient or with its electrochemical gradient from outside to inside, and that would release energy. And it's by creating these electrochemical gradients and then suddenly changing membrane permeability that neurons have the ability to send signals. To get a bit closer to understanding that, now let's look at two different types of membrane proteins that are drawn over here. Again, I'm drawing a membrane phospholipid bilayer, and this is outside of the cell, and this is inside of the cell. The first one that we need to consider is this one, and this is a ligand-gated ion channel. So when a ligand binds, this is a ligand, for example, a neurotransmitter, I'll write that as NT, then the channel opens. And as we've seen, if it's a sodium channel, then that will allow sodium to rush into the cell. Ligand-gated ion channels like these are present on the dendrites and cell bodies of neurons, so that if they receive neurotransmitter, they change their permeability. So for example, if it's a ligand-gated sodium channel, it would let sodium in. Sodium is positive. As it moves down its electrochemical gradient, it makes the inside of the neuron less negative and more positive. So let's go back up here to this oscilloscope tracing and see what that would look like. If the neuron is at its resting membrane potential, and then sodium moves in through one of these ligand-gated sodium channels, then the membrane potential would become less negative and more positive like this. We call that a depolarization. The membrane potential is polarized at -70, and now it's getting less negative, so that's depolarizing. So this is a depolarization. In contrast, a different ligand-gated ion channel might be a chloride channel. So I'm going to draw it on the same channel here just for ease, but imagine this is a different channel. So letting chloride in, and chloride is negative, and so that would make the inside of the neuron more negative, not more positive, and would take it down like this. The voltage would get more negative, and we would call that a hyperpolarization because it is getting even more polarized than it was before. Again, both of these can be achieved by ligand-gated ion channels, and because they can get bigger or smaller, more neurotransmitter will open more channels and make this bigger, or less neurotransmitter will open fewer channels and make it smaller. These are called graded potentials. So graded potentials are caused by the movement of ions through these ligand-gated ion channels. And again, this is a hyperpolarization. Now, if enough depolarization happens that the membrane gets to what is called threshold potential. Now, I've drawn this very much not to scale, but that's often about -55 millivolts. And this is threshold potential here. We'll label that 4. Then something very special happens, and another group of ion channels open, and those are called voltage-gated ion channels. So this is an example of a voltage-gated sodium channel, whereas this one over here was a ligand-gated sodium channel. And what's special about voltage-gated sodium channels is that they open, you can imagine this swinging open, in response to depolarization. I'm going to draw these little symbols here to indicate that the membrane around them has depolarized. That makes them open, and then sodium rushes in. That is actually a positive feedback cycle, because what we've got is depolarization causing more positive ions moving in, which itself causes depolarization. That runaway positive feedback cycle causes more and more sodium to enter the neuron, causes a greater and greater spike in positivity, and that's called the action potential, and shown on the oscilloscope here as this big positive spike. We're going to look in the next lesson in more detail at everything that's happening during the action potential. This one's just meant to provide an introduction to the terminology and to help you understand membrane potential. But just to finish the picture, after this initial spike caused by an influx of sodium ions, the second gate on the voltage-gated sodium channels close, and potassium leaves the cell through voltage-gated potassium channels. The combined actions of those bring the membrane potential back down. In fact, it gets more negative than resting membrane potential. It overshoots, and then it comes back to resting membrane potential, and we call this repolarization. So this is repolarization over here. Again, in the next lesson, we'll look in more detail at the action potential, but here you've seen an introduction to membrane potential and the electrochemical gradient, as well as the membrane proteins that maintain them.